Hello everyone, today we'll be discussing an important topic which is mutations. And mutations is very commonly asked in most of the university exam question papers. Now let us see what are these mutations. You all know that DNA is a carrier of genetic information, right? But if there is any change in the nucleotide sequence of DNA, that is referred to as mutations. So what are the different types of mutations and what are the different effects of mutations is what we are going to discuss in today's session. So there are different types of mutations, which is the common ones are point mutations, frame shift mutations and suppressor mutations. So point mutations are the most common type of mutations, and that is what we are going to discuss in detail in this session. So what is point mutation? If there is a change in the single nucleotide base of the DNA, that is referred to as point mutation. You can either add a nucleotide, you can either delete a nucleotide, or you can either substitute one nucleotide for another. All these three comes under the group of point mutations. So now let me show you with the example. So this is a normal DNA strand which is present, which is inherited from parents. Okay, so you have A. This is the sequence of the DNA strand. So for that you have the mRNA which was produced and you have the particular amino acid which is derived from that mRNA. Now you see substitution. So here in this sequence, in the sequence of DNA, here instead of this particular nucleotide, you have added another nucleotide. Instead of C, you have added T. Okay, so C is for cytosine, T is for thymine. So you have substituted one nucleotide instead of another nucleotide. So that is substitution. So as a result, what happens? See, the mRNA information is also changed and a different amino acid is added at that level. Okay, so that is substitution. Then you have insertion. So you see the same sequence is here. Compare it with the normal one. Okay, you have ATG is there. Here instead of there it was CTT, and here you have inserted another C, and the CTT is moved this side. So you have C, and a C or cytosine residue is inserted here, and that also will change the mRNA code, and that will also add a different amino acid. Okay, then you have deletion. See, for example, after AAA you had ATG, that A is removed. So you remove the A. There again, if you see, it reads TGC, and that the mRNA genetic code will also be changed. So after that you will be adding all different amino acids. So because the reading frame gets changed here, you know, the mRNA has to be read in a particular sequence for each amino acid to be added in that sequence and the protein to be produced. So now you see a small change in the DNA can lead to massive changes or abnormal proteins which are being produced, and that is the effect of mutations. This is more specifically about point mutations. Now let me introduce two different words, transition and transversion. Okay, so transition, if you replace a purine for a purine and a pyrimidine for a pyrimidine, it is transition. Whereas if you replace a purine, if the mutation is causing the replacement of a purine by a pyrimidine or a pyrimidine by a purine, then that is transversion. Okay, so these are the different terminologies you need to know with reference to point mutations. Now let me tell you, there is a classification of point mutations based upon the effects of the protein which they produce. They can be either silent mutations or missense mutations, or they can be nonsense mutations. Let me tell you with an example what are these. So now you have the codon here. For that codon you have the amino acid. Now let me tell you the different types of mutations. You had silent, nonsense and missense. So what is a silent mutation? See, here you have the codon GAA. It coded for the amino acid glutamic acid. Whereas there is one particular change. Instead of A, there was a mutation and caused G. Okay, so G was added. But anyway, you had only glutamate. I already told you, one amino acid can have more than one codon. So you are adding the same amino acid even if the codon has been changed. So what happens? There won't be any effector. And that's why it is referred to as silent mutation. Whereas, see the next one. You had GAA. That coded for glutamate. In that place, instead of G, you replaced a U. What happened? It became UAA. I told you UAA is a stop codon. So it will lead to termination of the protein synthesis. So protein synthesis abnormally stops or prematurely stops. and that is why it is referred to as nonsense mutation. And then finally see this one. You had GAA, which is for glutamate. If the last one is replaced by a C, it codes for a different amino acid aspartate. So this is a missense mutation. Okay, missense. So instead of one amino acid, you are adding another amino acid because of one single nucleotide change, and that is missense mutation. So these are the different types of point mutation based upon the effects. Now, under missense mutation, you have three different types of point mutations, which is acceptable, partially acceptable, and which is unacceptable or non-acceptable mutation. So what is this acceptable missense mutation? Now let me tell you with an example. Always we'll see with relation to hemoglobin. Okay, so in hemoglobin A, in this beta chain, okay, in the beta chain, in the 61st position, you have a lysine amino acid. If it is changed into an asparagine residue because of the mutation, you will produce hemoglobin Hikari. Okay, hemoglobin Hikari, which will not have much effects on the function of hemoglobin. So it is referred to as acceptable missense. It is just a variant of the normal hemoglobin. It will not have any effects on the function. So the oxygen carrying capacity of hemoglobin is maintained. So you're not worried about the mutation. And that is hemoglobin Hikari, which is an acceptable missense mutation. Here if you see, the codons have been, what were the different codon change has been mentioned here, which led to replacement of lysine in the 61st position by asparagine, and that is hemoglobin Hikari. You can accept it. It is not a problem. Then you have the partially acceptable, which is HBS or the sickle hemoglobin. Okay, here if you see in the beta chain in the 6th position, instead of glutamate, you have the amino acid valine. Instead of glutamate, you have the amino acid valine. This is partially acceptable. The reason being, sickle hemoglobin can remain quiet and not cause any symptoms also. Whereas only when the person or the patient gets exposed to hypoxic conditions, the sickling will be increased, and that leads to various crises, which is seen in sickle cell anemia. So that is the reason this is a partially acceptable missense mutation. And then you have totally unacceptable missense mutation, which is hemoglobin M or the methemoglobin, or the otherwise referred to as Boston hemoglobin. And here in the alpha chain in the 58th position, the histidine amino acid is replaced to tyrosine. So as a result, this HBM cannot carry oxygen effectively, and presence of HBM can lead to the development of cyanosis. So the function of hemoglobin is to carry oxygen effectively. The function itself is affected. Here it cannot carry oxygen, and it is so totally unacceptable missense mutation. Now coming to the last two sets of mutations, which is frame shift and suppressor mutation. And any insertion or deletion of more than one nucleotide will lead to a frame shift. That is, the reading frame of that, the mRNA will be read by the ribosome. That reading frame itself gets shifted. This results in a garbled appearance of the mRNA, and that may cause premature termination also. So here if you see, this is the original DNA which codes for an amino acid sequence. This is the amino acid sequence which is added here. Now, frame shift of one DNA base will result in a totally abnormal amino acid sequence. So here the amino acid sequence was histidine, serine, histidine, val. Here totally the amino acid sequence has been changed, which can lead to dysfunctional protein. That is about frame shift mutation. And then finally a word on suppressor mutations, which is the alteration in the anticodon regions of tRNA. So tRNA suppressor molecules, these suppress the nonsense mutations that occur elsewhere in the protein coding genes. So this is just for completion's sake. So mutations are nothing but they are any change in the nucleotide sequence of DNA. So here you need to know there are three types of mutations: point mutations, and then you have the frame shift mutations, then you have the suppressor mutations. In point mutations, we have seen there is a change in the single nucleotide. You can either add it, you can either delete it, you can either substitute it. Then we saw two different terms, transition and transversion. Then we saw about the different types of point mutation based upon their effects: silent, missense and nonsense. And in this missense mutation, we saw three types: acceptable, partially acceptable and unacceptable. Then we saw about the frame shift mutations and suppressor mutations.